My name is Herman, and today I will present the project called Green One-Pot Synthesis of Gold Nanoparticles as a prompt polyphenol assay and theranostic platform. First, we have the abstract where we talk about the gold nanoparticles. Gold nanoparticles have emerged as versatile tools in biomedical engineering, offering unique optical, electronic, and surface chemistry properties that enable applications in targeted imaging, drug delivery, and photothermal therapy. Their plasmonic resonance and high surface-to-volume ratio make them ideal scaffolds for multifunctional platforms in precision medicine. Meanwhile, polyphenol-rich extracts, particularly from pecan nut, are gaining attention as complementary agents in cancer treatment, showing promise in leukemia models through antioxidant activity and pro-apoptotic pathways. To unlock their full therapeutic potential, rapid and accurate quantification of total polyphenol content across diverse sample types is essential. The following speculative assay, or FC assay, remains the standard for total polyphenol measurements, yet it demands dilution of FC reagent, preparation of 1 molar sodium carbonate, and requires 2 sequential incubation steps, post-carbonate addition and post-reagent introduction, cumulatively extending analysis time despite shorter individual durations to develop a weak absorbance peak near 765 nanometers. This multi-step approach is time-consuming, reagent-intensive, and prone to interferences, limiting throughput in research. Here, we present a green method in which any polyphenolic sample, whether plant extract, byproducts, or purified compounds, is simply added to an aqueous solution of tetrachloruric acid and heated in a water bath. In minutes, bioactive polyphenols reduce ion golds to elemental gold, forming stable monodispersed gold nanoparticles whose surface plasmonic resonance provides a direct quantitative readout of total polyphenol concentration. We prepare serial dilutions of methanolic pecan extract from 0.8 to 0.0125 milligrams per milliliter in triplicate and generate calibration curves both for the gold nanoparticles method, which absorbance is at 522 nanometers, and the FC assay, which absorbance is at 765 nanometers. FC assay exhibited good linearity, but the gold nanoparticles assay displayed a significantly broader dynamic range of absorbance units, as it exhibited linear behavior at very low concentration range, but eventually reached a saturation point. A scanning electron microscope confirms the generation of uniform spherical nanoparticles, and Fourier transform infrared spectroscopy verifies retention of key antioxidant moieties on the gold nanoparticle surface. The resulting bioconjugates combine rapid polyphenol quantification with intrinsic reactive oxygen species suppression and plasmonic properties suited for targeted imaging or photothermal applications. Well, next, in the introduction, we have that polyphenols are organic compounds characterized by multiple phenol units. Polyphenols in the biomedical field are mainly used for their antioxidant, anti-inflammatory, anti-cancer, and neuroprotective properties. They help to protect cells against oxidative damage, modulate inflammatory processes, and may contribute to the prevention and treatment of chronic diseases such as cancer, cardiovascular disease, and neurodegenerative diseases. The quantification of polyphenols is important because it allows the concentration and efficacy of these compounds in biological samples or natural products to be evaluated, ensuring their therapeutic potential and aiding in the development of polyphenol-based drugs, supplements, or therapies. Polyphenols act as a natural reducing agents in the synthesis of gold nanoparticles due to their abundant hydroxyl groups in their aromatic rings. These hydroxyl groups can donate electrons to reduce gold ions to elemental gold, enabling the formation of gold nanoparticles. Additionally, polyphenols can stabilize the nanoparticles by keeping their surface, preventing aggregation. The synthesis of gold nanoparticles from polyphenols allows us to obtain 2 applications: the quantification of polyphenols and a theranostic platform. A theranostic platform is a smart nanosystem able to diagnose bioactive delivery and monitoring of therapeutic response. Well, the methodology used is confirmed by 4 steps. First, we have the preparation of methanolic pecan extract, in which 100% methanol was diluted to 80%, and then 40 milligrams of the polyphenol extract was placed in a volumetric flask and filled to 10 milliliters with 80% methanol, obtaining a concentration of 4 milligrams per milliliter. The next step was the serial dilutions of methanolic pecan extract. First, 1,000 microliters of distilled water were placed in 7 containers, and 600 microliters were added to the first container. Then, 400 microliters of the 4 milligrams per milliliter methanolic pecan extract solution was placed in the containers. Later, serial dilutions of 1 to 2 were performed in the remaining containers, taking 1,000 microliters from the first 2 and placing it in the next one. The previous step was repeated for each container. The series step was the deposition of tetrachloroauric acid in diluted series. 250 microliters of tetrachloroauric acid were added to each dilution. The final step was the synthesis of the gold nanoparticles by water bath heating. The containers were placed inside a beaker to which water was added and placed in a hot plate. And then the beaker with the containers was heated to high temperature for 15 to 20 minutes until the synthesis of gold nanoparticles was observed. After this, we obtained these results. First, with the scanning electron microscopy, or CEM, we observed that nanoparticles have a spherical morphology, and the size of the nanoparticles increases as the concentration of polyphenol extract decreases. Then, with the Fourier transform infrared spectroscopy, we observed that in the FTIR of the extract, the functional groups OH, ester bonds that are double bonds between carbon and oxygen, and double bonds of carbon, characteristic of the chemical structure of polyphenols, are observed. After that, in the FTR of gold nanoparticles, the functional groups mentioned above, corresponding to polyphenols, are also observed. Finally, in the UVB absorbance spectrum, in both spectra, the change in absorbance is observed at each concentration of the serial dilutions, and in the FCSA calibration curve, linear behavior is observed, while the gold nanoparticles calibration curve shows linear behavior at very low concentration as it reaches a saturation point. With these results, in conclusion, we observe that the gold nanoparticles calibration curve demonstrates linearity at low concentrations before plateauing, while FTIR and SEM analysis confirm the presence of functional groups on the nanoparticle surface, corresponding to polyphenol functional groups, and reveal that higher polyphenol extract concentrations produce smaller particle sizes. These findings highlight the method's potential for polyphenol quantification and the production of biocompatible materials suitable for theranostic applications. Thanks for your attention.